Actually, it was better to start our first presentation with this topic because it's how our work starts. 4 years ago, we got the idea of nanotechnology, and we start to hear about this in Egypt. And we start to investigate what is the biomedical application can be used for using nanotechnology. And according to my interest, I'm working in microbiology, so we start to work in antibacterial effect, antiviral effect, anti-cancer effect. But it was antibacterial effect of nanoparticle was well investigated by many researchers, and they're speaking about this, about ability and the potency of nanosilver for inhibiting of bacteria and viruses. Then we got the viruses. So but there was no any article related to Bovine Viral Diarrhea Virus or hepatitis C virus. Nothing. And actually, to work with nanosilver on this field is actually practically impossible because of toxicity. It's very toxic, highly toxic. So we start to work in this area, antiviral effect of nanoparticles. Here I am speaking about BVD virus as a model for hepatitis C virus. And we work in this virus using nanogold. But at the beginning, we start our work with many nanomaterials. First, we have some information about hepatitis C virus. It's a member of Hep C virus, genus family Flaviviridae. It's the same family of Bovine Viral Diarrhea Virus. Major cause of human hepatitis throughout the World Health Organization estimates that 150 to 170 million people chronically infected with hepatitis C virus. And I think in Egypt we have 50% of this ratio may be in Egypt. I have two in my family, my mother and my younger brother, infected with hepatitis C virus. People with chronic infection have the risk of developing liver cirrhosis and hepatocellular carcinoma. People with hepatitis C virus are asymptomatic until cirrhosis of liver occurs. Structure of hepatitis C virus almost the same as bovine viral diarrhea virus. Single-strand RNA nucleocapsid, E2 receptor, E1 and envelope glycoprotein. Limitation of experimental research on hepatitis C virus that it's impossible to propagate hepatitis C virus in tissue culture. No one till now can isolate hepatitis C virus in tissue culture. And also no experimental model, except chimpanzee, which is very difficult and very expensive and unavailable to have it. Limitation of current therapies of hepatitis C virus. They were using interferon, gamma interferon, and ribavirin, and nowadays they start to use Sovaldi. But all of these medications, still now, are very expensive, effective only in subset of patients, according to genotype of infection, and associated with many, many side effects, depression, flu-like symptoms, fatigue, headache, insomnia, hemolytic anemia. And actually, sometimes they stop medication. Why BVD virus? Within the same family, they are members in the same family. They are antigenically related to each other. They are biologically and genetic, they are the same in their replication cycle, biology, genetic organization. So functionally homologous nature of many of their genes products. Utilize the same receptor, LDL receptor to enter cell. Use functionally similar internal ribosome entry site for translation. Have mechanistically similar non-structure 5' protein RNA-dependent RNA polymerase and equivalent mechanism of viral maturation and assembly and egress. So we choose BVD to be our model for hepatitis C virus. Nanotechnology, an idea of nanotechnology is part of science and technology manipulation of the matter as atomic and the molecular size. They discovered when they reduced metal or matter into nano size, it acquired new properties. For example, we know gold with gram is something, and with nano size is something else. Totally different characters. And actually, this character can change according to size of nano, according to shape of nano, according to stabilizing agent, according to charge of nano. So we speak about billions of new properties we got in this science. This photo shows to us what is size of nano actually. The size of nano is 1 power minus 9 per meter. For example, this is bacterial cell, 10 power 3. This is virus around 10 power 2. And this is antibody around 10 nanometer. We speak about nano start from this area to this area. So we speak about particles less than size of antibody. With totally new character, no one, not full, we don't have full information about their new properties, their new characters. So can be made, also nanoparticles can be made of metals, semiconductors, oxide, polymers, particular, and they have new electrical, mechanical, magnetic, optical, chemical, other properties. And their properties are vary according to many factors as size and charge and shape and stabilizing agent used. Then actually after we got our results of inhibiting this bovine viral diarrhea virus with our nano gold 2012, I was unlucky that another two researchers discovered also the antiviral effect on HIV and also influenza virus. But already gold nanoparticles reported to possess in vitro antiviral properties. It was 2012. Aim of our work, in vitro evaluation of antiviral activity of gold nanoparticles on BVD virus as a model for hepatitis C virus. Of course, we didn't start with gold only. We started with gold and silver and copper, and we studied many sizes and many charges and many shapes. At the beginning, we were blind. We don't have any one reference. And the two references who work on HIV and influenza virus, actually they published the work after we already finished our work and we got our result. But we were unlucky to publish it so fast. Material and the methods. The same, we have also same cell culture, Madden Derby bovine kidney, supplemented with horse serum, 5% and incubated in 5% carbon dioxide incubator. Virus, BVD virus, needle strain. Gold nanoparticle, we prepare it ourselves. Colloidal gold nanoparticles inside optimization and classical citrate reduction method according to this process. Surface modification of gold nanoparticles, we used BEG, polyethylene glycol, which increase colloidal stability of our gold nanoparticles in physiological fluids, and also play a role in targeting of these viruses, of these nanoparticles, play a very important role in targeting of our nanoparticles. And then we make our characterizations using spectrophotometer and zeta sizer and transmission electron microscope. Before we start to study antiviral effect of these nanoparticles, we have to estimate the cytotoxicity of these nanoparticles, and we have to decide which concentration or minimum non-toxic concentration or safe concentration to be used. We use this technique, MTT assay, which is calorimetric assay reflecting cell viability. This picture, it's calorimetric assay in which cells transform this MTT reagent into tetrazolium insoluble salts. And this test actually reflects cell viability and cell metabolic activity. Increased color, meaning increased cell viability. From first assay, MTT assay, we got best concentration to be used in, and the safe concentration, non-toxic concentrations. And we start second step in our experiment with demonstration of antiviral activity in two ways. First way, we allow our nanoparticles to be in contact with the virus for 15 minutes prior to infection of Madden Derby bovine kidney cells. And we used 2 and 4 and 6 and 8 parts per million concentration of our nanoparticles. And this concentration as a concentration which possess very low toxicity. Second way, viral particles were first allowed to infect Madden Derby bovine kidney cells for 1 hour. Then we add our nanoparticles in order just to understand when exactly inhibition occur, before attachment or after attachment and entry. And of course, triple control and triple positive and negative control. Our results. Our particles, best result we got when particle size was around 7 nanometer. And the best charge was minus 12.3 millivolts. This is our particles, 7 nanometer. 9 nanometer. This is size range distribution of our particles. Mainly, they are 7 nanometer, I'm sorry. Size distribution by zeta sizer. This is charge of our particles, minus 12.3. This is our result of cytotoxicity assay. Here we have cell control, 0.28, 2 and 4 also same exactly. So we can say that 2 and 4 parts per million are non-toxic concentration, which I agree with many authors. Result of cytotoxicity assay, graph. Then antiviral. This is our virus group, cell control, virus control. Virus control was 0.11, where the cells completely destroyed. Cell control 0.27. And we have this concentration 4 parts per million, which completely inhibited the virus. This is graph to describe this, 4 parts per million equal to cell control. Our results are exactly agreeing with Summit Band and Kiskar et al., who say the same about our virus, but about HIV and influenza virus. But of course, not the same nanogold particles. Another nanogold particles with another characters, another modifications. So antiviral activity evaluation of prepared gold nanoparticles showed greater activity in the test where the viral particles were allowed to interact with nanoparticles before infecting using method 1, as compared to these cells previously infected with the virus method 2. Result confirmed that BVD virus used as a study, it's okay, 4 parts per million concentration were found to be effective as virus analysis candidate, which allowed to interact with cells. Actually, after we got this result, we start to work in vivo, to want to work in vivo inhibition, and we didn't find host for BVD virus. So we start to work on adaptation of this virus to be working in rabbits and to do in vivo experiments. And actually we did it, but results was not that good. So, and we are trying to improve it in the future. Thank you.
Time for several questions from the audience.
Any questions?
Will the nanoparticles cause damage to your body organs?
No.
No.
It varies according to type of nanoparticles. Do you understand me?
Yeah.
When we used silver nanoparticles, we didn't. It's very toxic and actually cause severe damage. And there is 4 years ago they discovered that it can really inhibit AIDS virus, HIV virus.
Yeah.
But practically they can't use it because it's very toxic. Yeah. So because of this, we start our work in nanogolds because it's much more safer. Yeah. And according to stabilizing agent used on this nanogold, because also this stabilizing agent affect charge and affect excretion and of this nanoparticle out of the body. So if we speak about nanogold virus, nanogold particles at certain sizes with special stabilizing agent, we can say that it's almost safe.
Yeah, because the field also moved to biodegradable nanoparticles because of this fear of the interfering damage
like
to the organs.
Like chitosan and also new polymers. It's totally new, but every one of these nanoparticles has different characters. For example, we can say that nanochitosan can inhibit the virus. No, it cannot.
Thank
you. The presentation. When you started your presentation, you said you tested for particles.
What?
When you started your presentation, you made mention that you tested for different nanoparticles.
Yes.
So why did you not prefer good particles, nanoparticles?
What?
Why did you not prefer good particles for your study?
Good particles?
Good.
Gold prefer because it's non-toxic, showing minimal toxicity. Okay. It's showing minimal toxicity. We start with silver, very toxic, copper, very toxic, but but nanogold is quite safe.
Okay, but what of iron? Iron oxide nanoparticles, is it toxic as well? Iron.
Iron?
Yeah.
We didn't try it. We didn't try it. Unfortunately, we didn't try it. We tried nanoparticle already mentioned to possess antibacterial activity.
Okay, because I know that on iron and titanium nanoparticles, they have so many advantages over good nanoparticles.
Oh, I can read about this. Thank you.
All right. Thank you.
There's another question.
Nanoparticle.
Yes? To detect the virus? Of course, it's very easy. Price of gold nanoparticles now, because it's still under research, it's quite expensive. But to prepare it yourself in your lab, it's very cheap. Very cheap, and doesn't need any big facility, just a magnetic router. That's it.